All right. Thank you, Rahul, for the kind introduction. And thanks for the opportunity to speak. It's my pleasure to speak here. So my talk, like Rahul mentioned, is focused on MetID, but the impact of what I will show here is way beyond that, but we'll try to keep it focused to the MetID. We've used the fragmentation modes, the CID, EAD in conjunction, and we've also compared the 7600 and the 8600 in partnership with SCIEX. So I'm going to get to the very why do we care, right? The why. Start with the why. So we care because we want to understand the human metabolism. And we're a pharmaceutical company. We develop xenobiotics. ADME is key to understanding how these work. And we can break this down into 4 parts. I take my medicine, it absorbs in the body. How does it go in? It distributes in the body. That's where one of our other team, the disposition team, comes in, where we do mass spectrometry imaging to understand where this stuff is going. Then we have to understand what happens to it, because if it sticks around for too long after doing what it's supposed to do, it turns toxic. So we don't want that. We want the drug cleared. We want it cleared in a reasonable amount of time. We want to achieve the mass balance. We want to get 90% plus back. We do that by radioactive studies, so I'll show you that in a little bit. And then excrete, right? So we will test out the excreta to get all that mass balance data. So circulating drug-related material is key. Identify and quantify. We want to identify all the major metabolites. We want to identify the minor metabolites. We want to understand the nature of these. What is happening to these? Is there any tox liabilities? We want to understand the complete elimination of the drug. Once again, I'm reiterating these messages. And then we want to be able to take all this, and in our case, we want to get the safety coverage. That's the key. So metabolites and safety testing, missed. So let's go, and I'll give you a brief overview of what our team is. Again, drug metabolism, these are core areas. So we impact all the way from early discovery all the way to the actual clinical samples, which is the first in human studies, tox studies, as well as the key human ADME study. And we will influence, early on, we'll influence the sort of drug design, how the molecule is shaped by identifying soft spots, modify the chemistry, and later on, we do all the other things. And the data that we provide is part of the regulatory filing, so we cannot miss a metabolite. At least we try not to. I mean, mistakes will happen. We're human. But here we go. So this is the entire pharma pipeline, and if you go to any pharma talk, a lot of people like to show the pipeline, so here I am showing the pipeline. So we have our discovery, which is the top left, since I do not have a laser pointer. So look at the top left corner, where we start with the candidate nomination, candidate selection. That's where the chemistry happens, right? So you have your tier 1, tier 2 assays. All of these are happening in our shop, and all of them we will be doing we're doing them with the 7600 right now, and hopefully we'll add on the 8600 and its capabilities. So the tier 1, the exploratory in vivo, the cross-species hepatocytes. This is a key study pre-development, because this helps us identify which species to pick. Because if you see a liability in the human ADME, so now let's go down to the development portion, where the human ADME study is the sort of done during the phase 2 of development. We're trying to create a human-first approach, and that's a whole different talk of trying to do these studies early. But the key studies there are the first in human and human ADME studies. The previous studies all guide those studies. And if you have a finding in a human study, let's say you get a major metabolite, which is 10% of drug-related material, you want to have coverage in a preclinical species. So either in a rat, which could be your rodent ADME, and you also have your non-rodent ADME, which is a dog or a monkey. And you want to make sure that that metabolite is tested and it's covered. By covered, I mean you see it's a match. So you have the same exposure or more in your preclinical species. Then you have proof that it's safe and it doesn't warrant. Otherwise, imagine triggering something at a late stage and slowing everything down. That's not ideal. So now I'm going to jump into the pathways of drug metabolism, the phase 1 and phase 2. So you have your functionalization of your molecule, that's your phase 1. Everything that is happening here is so that you can excrete your drug, whether you pee it out or poop it out. Yeah, so you've got everything that's happening, phase 1, phase 2. Phase 1 is functionalization, phase 2 is conjugation. I'm not going to go into the list, but if you're a mass spec person, you like those, decimal points ahead, especially in the high-res space. That's what we're looking for. Those are the different. We're also a group where we have several organic chemists, and I'm not an organic chemist. I'm not an expert in organic. I'm still learning as things go, but we have to understand organic chemistry. What is happening to the molecule? Where is it being modified? How does the conjugation add on to it? And this is a puzzle. That's literally what I want to show on this slide, is that you can attack a puzzle using many different strategies. And these fragmentation modes are your different ways of solving a puzzle. Now, an instrument like the 7600 really gives you multiple tools to solve that puzzle. You're not just one-dimensional. So you have, within your one run, you can set up smartly and solve the puzzle better than other platforms. Now, again, these are the 4 things I just talked about. I've made simple cartoons, which, you have your CID, you have your HCD. Now, your HCD, which is your high energy collision dissociation that happens on the Orbi platform, and the CID that happens on your 7600, we happen to have both, are extremely, extremely comparable. So your CID matches that. And then you get this bonus EAD fragmentation. It's like buy one, get one free. And it literally, it works for us that way. So I'm going to show a few examples. Like I said, I'm not going to go deep into the organic chemistry here, but I've shown the different cleavage points. This is a trisebian phosphate. And the point I'm trying to make here, since I don't have a laser pointer, this slide is going to be quite irritating for people. So just look at the EAD and look at the blue stars. And those are the unique peaks that you start catching in your EAD. Now, if you're trying to solve a puzzle, which some of us are, and trying to identify, the more things you have to look at from your fragmentation, the better it gets, the higher the confidence you get. Now, ultimately, NMR is a gold standard. No amount of mass spec is going to you would have to isolate. If you want to get an absolute structure on something, get NMR and get X-ray. But this tool gets you pretty far along to where you need to be. Then we're going to go into one of the clozapine. I'm going to talk about the clozapine a little bit more. Again, you see a unique peak at 282, which is kind of allows us to solve where this, the structure a little bit better. And the same thing for your glucuronide. You see 2 extra peaks there. Again, the 2 blue stars that I was talking about in the bottom EAD spectrum that are unique. And we've also compared with UVPD, and in some cases, UVPD does provide us with useful information as well. But EAD, generally, we've seen a little bit more, actually quite a bit more peaks showing up in the EAD than in our UVPD workflows. So we want to jump straight into our discovery studies, where we were benchmarking sort of the 2 platforms. So we shifted to SCIEX now for doing our discovery studies, because of the speed of the time of flight platform, and we've been able to speed up all of our assays. This is our sort of clozapine data for dog on platform 1, and here I'm showing the equivalence data. Our goal is to reduce this time to like 5 minutes or under for all of our screens. So we do S9s, we do, like I said, we'll do chromosomes, we'll do cross-species hepatocytes, a lot of these different rat liver microsomes, not chromosomes, rat liver microsomes. And we basically get all of these to get our compounds turned over. The dog and human data match pretty good. I'm not jumping into this. I'm just going to go straight into what we did. So we utilized the speed of the TOF. We automated our data acquisition in 3 different modes. That's the other beauty, which I did mention. So the SCIEX instrument has the mass defect filter and isotope pattern recognition up front. Most other softwares offer this at the back end. So we've actually acquired data using these different workflows, and it really, really gives you a sort of filtered data. So it's like you're filtering your data up front rather than post-acquisition. We can do quick and agile studies, and again, I'm not going to go into the laundry list, but this is where we kind of want to identify our soft spots and give the chemists some really good information to work with and design better molecules. This slide was already shown, so here I'm going to pivot and actually talk about my own journey with SCIEX. And like Rahul said, I've been in the industry for 17 years. I'm almost as old as the 5500. And both of us are still working. So, I mean, the 5500 is, I think, in my opinion, a testament to a robust and classy platform. So I'm going to start with where I started with SCIEX. It was a Q-Star Elite, and it had a MALDI source. I don't know, there were only 8 made, so I would be shocked if anybody else has used it in this room. But then we had a 5500, and the one I had was a Q-Trap. So I came from Purdue, from Professor Graham Cooks's lab, and we put a DESI on that. And we did a lot of work using our 5500 platform. We made a few friends along the way. I would give a shout out to Kim Redlick, who's no longer with us. He was our service engineer. I shamelessly custom modified the front end, and he never gave me trouble about it because I was heating it way more than it was allowed. That's what allowed for the ambient stuff to happen. We fitted it with paper spray. We fitted it with low temperature plasma. We even put an AP MALDI source on it at some point. Then we moved on to the 6500. We've also used the 7500, then the 7600, and now we're working with Rahul and SCIEX in partnering on using the 8600. So it's been quite a journey. Made a few friends along the way, Spencer, Rahul, Randy. A shout out to all of you who've been part of this journey with me. So, I mean, really, SCIEX has been a great scientific partner to AbbVie. And I'm going to jump into this sort of benchmarking the 8600 with the 7600 comparison now. This list is by no means a complete list of what we've identified. We've just taken 4 examples from each, and I'm going to go into what kind of enhancement in the 8600 signal we're getting and why that's significant for us. And at the end, I'll hopefully tie all the loose ends together and tell you why this matters. So, jumping into the first example, the clozapine glucuronide. Here we were seeing a freakishly higher signal. Rahul and I are still trying to understand and go deeper into. This was a very nice improvement from your. So the data on the top is your 8600, and the data at the bottom is your 7600. So just look for the counts per second. That's the sort of emphasis. And this is, you have your MS, and then you have your MRM-HR scan. And that's where we are seeing a boost in the sort of sensitivity. And I'll put it in a table later so it's easy to understand. So this is example 1, pretty significant boost. Example 2, another metabolite with clozapine. We see, again, 200 counts and 14 counts. So things that are actually hidden in the noise, and that's what we've just shown a few minutes back, are beginning to pop up. And for somebody who is elucidating the structures and looking for more signal to noise and more confidence in trying to identify what's out there, this data is very, very valuable. And both MS and MSMS are enhanced pretty significantly. Then you move on to another example, which is the glucuronide, the tamoxifen. Same thing. You see your counts go up, not that much, but you see a pretty decent increase. It's across the board. And the key here is to find these low-level fragments, right, that are unique. So if anyone who's done EAD, it's a mindset. When I first saw the EAD, I was like, what is this? This looks like if you're classically trained in mass spec, you're looking for that 10% of your parent and then look for the fragments. EAD fragments a little differently. So you have to get used to that. But once you get used to it, especially with this 8600 platform, man, it's the signal to noise is beautiful on the fragments. Again, another glucuronide. Glucuronides are very important to us because they tend to just fall apart, and we try to identify the exact site of localization. So any extra peak is amazing for us. And then finally, Piritabevir, and that's your last example. So that's a tongue twister. Anyway, now I'll go to two slides that matter. Here we see, so exactly like Elliot was showing, we have 30x increase in your MS signal across the board. It varies, and we also have the 13 times MS improvement in signal. And this is again, early work. It's sort of hot off the press kind of thing. We're going deeper and deeper into this. This thing is just launched, so give us a break. I'm just saying, yeah. But at least the beauty is it's across the board. I mean, that's what's promising. Now, why do we care if this happens? The reason we care, so I'm going to go over what we just showed. So we did CID, we did EAD. We know that it does as good as the HCD, and the EAD is outperforming UVPD. EAD yields unique fragment ions. When we jump to the 8600, this really becomes valuable because your signal to noise is enhanced. And we're applying this straight up to all of our animal and human ADME studies going forward. We have a few FIH studies in-house. Those are run without radioactivity. So every ounce of sensitivity you can pull out, it's going to be very, very, very valuable for us. So we have several of those lined up at AbbVie. We also benchmark the two systems across the board, MS and MSMS improvements. Bottom line is this study, showed us that this increased sensitivity for all of our human ADME studies where we dose low, sometimes you have tracer doses. You have, especially in neuroscience, if you're dosing in low amounts, what you get back and the amount of plasma given to you is finite and limited. So when you have dosed low and your sample limited, double whammy, if you have a platform that is 10x boosted, you inject 10 times less. If anybody who's worked in MetID, they know injecting 4 mils on a column is not fun because you're fraction collecting. So instead of 4 mils, imagine injecting 400 microliters. And that's where the power of this, I think, lies for us. It is going to impact key human studies going forward. So thank you for your attention. I am more than happy to take any questions you might have.